Right, so this is the biology combined, so it's trilogy, higher paper, paper 1. Now, question number 1. Now what I've done through this video is I've just split it up into every single question. All right, and then what I'll do is I'll then talk about exactly what my train of thought is. So question 1, right, bacteria can cause a variety of different diseases in humans. Right, so my first thought is I'm then going to draw a bacteria, right, because of what I reckon is, right, I reckon that there's going to be something to do with the structure of it. So that's the cell membrane, no, cell wall, God, stupid, cell wall, that's the cell membrane, they are then flagella going around the outside. Now that looks like a nucleus the way I've drawn it, but that's a plasmid. That is then sort of DNA material that's in there, and then it's got cytoplasm, because a bacteria is a prokaryote, so it has no nucleus. Now let's read the question. What are two similarities between a bacteria and an animal? Right, animal cell. Right, there's an animal cell, so there's the nucleus, there's the cytoplasm, it's got ribosomes, it's got mitochondria, and it's got a cell membrane. Okay, two similarities between a bacterial cell and an animal cell. Tick 2 boxes. Complete and utter plonker if you don't tick 2 boxes. So both have a cell membrane. Yes, yes, tick. Both have a cell wall. No, an animal cell doesn't have a cell wall. Both have a nucleus. Bacteria don't have a nucleus because they're prokaryote. Both have a cytoplasm or cytoplasm. Yes, yes, tick. Just to check on the last one, both have plasmids. No, animal cell doesn't have plasmids. Question number 2. Salmonella food poisoning is caused by bacteria in food. Okay, so we're talking about bacteria again, right? We're talking about salmonella. Salmonella, nasty food poisoning. Okay, I've had salmonella food poisoning a few times. Not particularly very nice. Give one symptom of salmonella food poisoning. All right, so I'm just going to think about it now. I remember now I had vomiting and I had diarrhea. Okay, I've put 2 symptoms down, but they're both valid. Oh, do not refer to vomiting or diarrhea on your answer. Good job I read the question. Scribble them out. Okay, so fever, stomach cramps, sweats, chills. Okay, all those different ones could then be put down, right, as one symptom of salmonella. And what you do is you put the one that you think is most likely going to be the one that is correct first. 1.3, what is the name of the first antibiotic developed? Right, there's no logic to it, right? It is penicillin, right? That Fleming guy, I think, right? So what we've got is we've got penicillin, and that's one that you've just got to remember. Okay, it's just like a fact. Oh, a graph, cool. All right, so let's have a look at it. Right, before we even think about any questions at all, let's have a think. A child with severe bacterial, right, and antibiotics. Right, now we know that bacteria are affected by antibiotics, but viruses, protists, fungi aren't affected by antibiotics. Figure 1 shows how the concentration of live bacteria in the child's body changed when taking a course of antibiotics. Right, so we know that if you take antibiotics, it's going to get rid of the bacteria. So let's have a look at the graph. Time in days, so it goes up to what, 4.5 days. That is when the antibiotic was then started. Concentration of live bacteria in the body in arbitrary units. So arbitrary units kind of means just like a made-up unit. It's just kind of relative. And what it starts at is it starts there, which is about 3.2 arbitrary units. Now I'm looking at that. If I start the antibiotics, I reckon there's going to be something to do with the fact that it goes up. Why would it go up if you've just started taking your antibiotics? Don't get that. Okay. The concentration of live bacteria in the body continued to increase after starting the course of antibiotics. Right. So what it is actually doing there, it is actually asking me about that. Suggest one reason why. Live bacteria are still reproducing. Right. So if you think about it, at this point here, right, the bacteria is still reproducing. So it's going to take a little bit of time, right? In fact, it takes almost a day for the actual antibiotics to start killing more bacteria than are actually being reproduced. So you could put down there is a time delay, right? Because there's a time delay before you have it. All right, and they are then the two kind of main reasons, and it sort of makes logical sense. You take the antibiotic, it takes a little bit of time before it then starts. So if you had a headache, and what you did is you then took paracetamol, your headache could get a little bit worse because it's kind of getting worse, and then it then goes down. 1.5. After 3 days of taking the antibiotic, the child felt better. Fantastic. There were still bacteria in the child's body. Why did the child feel better? Well, if there's still bacteria in there, right, then there is kind of going to be a thing. So now if I think about bacteria, right, how do bacteria, before I read any question here, how do bacteria actually make me feel ill? What they do is they produce toxins, right? And it's those toxins that are actually making me feel poorly. So let's have a look. Why did the child feel better? Tick one box, right? If you tick 2 boxes, you're a plonker, right? If you tick no boxes, you're also a plonker, because if you've got no idea, you might as well guess. Bacteria had become immune to the antibiotic. It is never, ever, ever, ever going to happen that quick. The child had become resistant to the bacteria. No, right? That's again not really possible for that to actually happen. There were fewer toxins in the body than in day 0. Correct. Question 1.6. Suggest why doctors do not give antibiotics to patients with minor infections. Right, if you've got a minor infection, right, what's probably going to happen is you're probably going to get better anyway. So there's almost no point in actually even going to the doctors in the first place because you are going to get better. Also, a minor infection could be a virus, right? And if it is a virus, antibiotics aren't going to do anything anyway. So let's have a think about it. Suggest why doctors do not give antibiotics to patients with minor infections. Now, what we're going to do is we are going to prevent, so we don't give them to prevent resistance to antibiotics. So we don't give antibiotics. You only give antibiotics when you really need to, right? Because the more times you give it, the more chance that the bacteria are then going to become resistant to it. And if they become resistant to it, life gets harder. It's actually in the answer sheet here is you're probably going to get better anyway. And it also could be a virus. So then 3 possible answers that you could have. Right? Don't give antibiotics because number 1 is you don't want the bacteria to become resistant. You could get better anyway, and it could be a virus. Right, figure 2 shows blood viewed using a microscope. All right, blood. Right, let's think about blood. I'm going to do it in red. Right, I'm going to do what's in blood? Red blood cells. Okay, what are they? There's lots of them. Right, they're donut shaped, so they're like a disc. Right, predominantly more than most. What else have we got in it? We've got platelets. Okay, so what they are is they are fragments. So if they are a fragment, that probably tends to mean that they are small, and their job is to then clot blood. Job of the red blood cell, carry oxygen. What else have we got in here? We've got white blood cells. Okay, we're doing white, but it's got a white background. White blood cells, they tend to be bigger and not many, because most of the time you don't really need very many. And the final bit is plasma. Plasma is kind of like a straw-coloured liquid. right? And what it is, it's all the liquid kind of in between these different things. So if I look at these now, A is probably a red blood cell. B, bigger and not as many, white blood cell. C is the liquid in between, plasma. D, small little fragments, platelets. Right, so before I've even gone through the question, what I've then done is I've gone through my knowledge, right, of what is in blood. Right, a vaccine will stimulate the production of antibodies. Which part of blood in figure 2 produces antibodies? Well, it's not red blood cells, it's not plasma, it's not platelets. White blood cells are the sort of immune system. All right, so tick 1, B. Which part of the blood in figure 2 starts the clotting process? Okay, I know that already. Small fragments, D. Question number 2. This question is about cell division. Right, before I read anything else now, what I'm going to do is cell division. So cell division as a center, right, pie chart. That then there is interphase, that there is mitosis, that there is cytokinesis. Can't fit it in very well. So interphase is kind of where all the mitochondria, the ribosomes, the amount of DNA that's in a cell doubles, and it goes all the way around there. Mitosis is then where the actual DNA itself splits, right? There's that full process, isn't there, of what happens in mitosis. And cytoplasm is then when the whole thing splits into 2, and we get 2 different cells at the end. So let's have a look at the question. The question is about cell division. Okay, I've just done it over there. That's cell division. Write the biological structures from the box in the correct order of size. Oh, it's got nothing to do with that. Okay, so we've got cell, chromosome, gene, nucleus. So it's something to do with a cell. So what I'm going to do is I'm going to draw a cell, and I'm going to draw the nucleus in it. And then what I'm going to do is I'm going to look at the question, and it says there smallest, and it says there largest. Now, my logical brain says biggest first. Okay, so what I'm going to do is I'm going to start big. So which is the biggest one out of all these? I've just drawn it, a cell. Inside the cell, I've then got that there, the nucleus. Inside the nucleus, so if I then zoom in on the nucleus like that, what I've got is I've got those little strandy things. And what those strandy things are chromosomes. Chromosomes. If I then zoom in on a chromosome, each one of these little tiny bits on there is a gene. There you go, smallest to largest. Figure 3 shows how a fertilized egg cell can produce specialized cells. So specialized, right? So what we have is an animal cell, and a specialized cell is where they change to be able to do a specific role. Okay, they sort of they become different, right? So what they do is they become different to do a role. Right, so fertilized egg cell. Fertilized means we've had an egg, I don't know why I did that, and a sperm. They've then come together and we've got a fertilized egg cell. Then what it does, it starts to replicate. That is then when it becomes an embryo. Process A, now I'm pretty confident that what they're going to do is they're going to ask me what process A is. It could become a red blood cell, it could become a muscle cell, it could be a nerve cell. Right, so they're the 3 specialisms. Oh, surprise, surprise. Name process A. That is differentiation. Okay, so that is then the actual process that is then expected, is differentiation. How many cell divisions are needed to form a 16-cell embryo from the original fertilized egg? Right, there's a fertilized egg. And then what they do is they split. So when they split, I then get 2. So that is 1 cell division. Then what they do is they split again. So that's the second cell division. How many am I after? 16. Then they split again. So 4 then becomes 8, which is 3. 8 then becomes 16. 4 cell divisions. 2.4. In humans, a fertilized egg contains 23 pairs. Pairs means 2, so therefore if there's 23 pairs, there must be 46 chromosomes. All right? Fertilized egg, 23 from the egg, 23 from the sperm. Right? That's my thinking. How many chromosomes will there be in each embryo cell? So in one embryo cell, there's 23 pairs. That means there must be 46. Here we go. Here we go, cell cycle. Okay, I've just drawn it then. Stage 1, stage 2, well, they've named it mitosis in that one, stage 3. And that there is the start. Okay, so I drew it before, didn't I? So stage 1 is interphase, where all the ribosomes and mitochondria and the amount of DNA doubles. Mitosis is then where the nucleus kind of doubles, the DNA in it. And stage 3 is cytokinesis. Describe one change in the cell that occurs during each of the stages of the cell cycle. All right, so what do we think is going to happen in that? It's called interphase. Right, now that sounds posh and that sounds like I'm being really, really clever, right? But that isn't a change. All that is the name of it. So it's a bit irrelevant, really. So what actually happens in there is we have the subcellular parts increase, right? So your ribosomes, your mitochondria, your cytoplasm, all those kind of things increase. And what you can do is you can also put in there ribosomes increase, mitochondria increase, DNA increase. So what I've done then is I've done the name of it, where I actually don't get any marks for at all. So what I'm going to do is I'm actually going to scrub that out because that's rubbish. Subcellular parts increase, that's just like posh saying that ribosomes, mitochondria and the DNA increase. So that's stage 1. Stage 2, give one change in the cell that occurs during each of these stages. So what I've now got is I've now got a big cell, right, and within that I've got the nucleus, and in there the amount of DNA has doubled. So what's the first thing that happens is the chromosomes line up, because that's what they do. And eventually at the end of it, we've got 2 nuclei. Okay, so you could say they line up, they divide, they're producing 2 daughter cells or nuclei. Stage 3. Stage 3, there, all right? Now, I know that is called cytokinesis. Again, you're not going to get the mark for that, okay, but there is absolutely no harm in writing it down just in case it's in the mark scheme. But this does kind of explain it a little bit. It's cell move, cytokinesis. So with that one, what you actually see is you'll see divide into 2 cells. And what are they? They are identical. Question 2.6. Cell division is important in the growth of multicellular organisms. Growth. Okay, so growth, that's kind of what we're thinking about here. Figure 5 shows the mean height of boys and of girls from birth to 18. Right, let's have a look at our graph. So it starts at 0, so that's how long all the babies are, and it ends at 18, which is how long all the 18-year-olds are. Mean height. Okay, now what I can do there is I can see I've got dots for boys. There's the boys, do them in red, right, and girls, a straight line. So if I'm looking at that there, right, what I'm looking at is I'm looking at the dots and the lines look pretty much exactly the same until they get to about that point there. I'll do a different colour. Until they get to about that point there. And then there's kind of a little bit of a gap. Then I can see that what they do is they cross over there. Maybe that's going to be important. I can also see that the girls goes a lot steeper there. Maybe that's important. Then I can also see that the girls flatten out there. Maybe that's important. The boys, let's get the red out, cross over there. Maybe that's important. And the boys stop there. All right? So there are lots and lots of little different places on there where there's kind of little changes on the graph that might make you think that there's going to be something asked about. Right. Now, what I've done on this question here is I've put in the part of the mark scheme. And it says here, level 1, which is 1 to 3 marks, relevant features are identified and differences noted. So what that's going to do is it's going to be boys are taller than girls. Girls are taller than boys at the age of 12, whatever it may well be. Level 2 is when you're talking scientifically in the ways they are similar and different is made clear, right? So what you're doing here is you're looking at similar and different. And the magnitude means how much, right? So magnitude means a number. Right, here's the question. Compare the growth of boys and with the growth of girls. Right, so what we've got to do then is we've then got to think about compare. If you're going to compare, you've got to talk about boys and girls in that same sort of fact at the same time. Right, so I'm going to say at birth, boys, now whenever you're doing your exam, you obviously don't do it short like this. Boys and girls are about the same, okay, height. Okay, so boys and girls at birth are about the same height. It stays like that up to age 11, up to age 11, boys and girls are the same height. Okay, then what we've got is we've got a bit of a change. 11 to 14, girls are taller, right? So if you think about that diagram I had on the screen before, right, the graph, what I'm doing is I'm just highlighting all these things. So 11 to 14, girls are then taller, right? They cross over, don't they? So when they cross over, that was at the age of 14. Boys and girls are the same mean height. All right, let's look at this now. There's 6 marks, and I've just done 1, 2, 3, 4 already. Older than 14, boys are taller. Boys' mean is higher. Right? So what I've done there is I've just done lots of observations of it all. Now, I was saying 4, I've got 1, 2, 3, 4, 5, 6, right? Now, that's fantastic, right? What I've done there is I've done 6 observations. Now, if you want to then start aiming for those better grades or better numbers, you need to think a little bit kind of more out of the box, right? So rather than just saying an individual fact, we then maybe need to talk about rates. Okay, so from the age 10 to 14, girls grow faster. Okay, so where in the previous one, what I've just done is I've just done facts, they are the same, they are taller, they are these. Now what we're doing is we're talking about the rate. At the age of 10 and 11, girls have a spurt. At the age of 13, 14, boys have a spurt. Okay, so the difference in this now is the fact that I've then got this as a rate and I've got these then as individual facts. 2.7. Give one way that cell division by mitosis is important in fully grown animals. All right, so here's me. That's obviously me, right? You can tell because the way my eyes are, right, and the size of my ears. Right. Anyway, probably shouldn't really be doing that in an exam. Now, mitosis is growth and repair. Okay, growth and repair. Now obviously at my age I'm not really growing anymore. I might be growing sideways, okay, but that's not necessarily the way that I'm going to be thinking about this. So mitosis is growth and repair, and because that is then highlighted as fully grown, it's not going to be that. So what I'm looking now at, I'm looking at repairing cells or replacing cells, and it is not growth. Question number 3. Amylase is an enzyme that digests starch. Warning bells, required, practical. Okay, I already know it's going to be something to do with the required practical because we're talking about amylase. We know that's an enzyme. Digest starch, we've done it all before. So what we've got is we've got our spotting tile. Okay, so all I'm doing here now is I'm just thinking about spotting tile. What are they going to be asking me about? Are they going to be asking me about pH? Are they going to be asking me about temperature? Because they could be any of the kind of questions that they're then asked about. The question, which organs in the human digestive system produce amylase? All right, nothing to do with required practical. So what I do is I draw me again. There's me. Okay, smiling. Okay, you've got one big eye and one little eye, but I guess that's irrelevant. Where do I produce amylase? I know that I produce amylase in my saliva. So what I'm going to do is I'm going to say mouth. Okay, then it goes down into my stomach. What do I produce in my stomach? I actually produce pepsin. All right, so it's pepsin, that's not an amylase. It then comes out of my stomach into the small intestine, and the small intestine does produce stuff like amylase, right? It produces lots of different enzymes. Around there as well, I've got my liver and I've got my pancreas. Liver produces bile, so it's not liver, it's not stomach. Pancreas produces amylases, proteases, lipases. That's in my pancreas. Right, so it's in my mouth, it's in my pancreas, and it's in my small intestine. Is there anywhere else? Then it goes from the small intestine, it goes into the large intestine. Large intestine doesn't do any digestion, all it does is absorb water, and then it comes out. Right, so let's have a look. Which organs in the human digestive system produce amylase? Right, so I've already decided it's mouth, I've decided it's pancreas, and I've decided it's small intestine. Liver, nope, that doesn't produce it, that just produces bile. Large intestine, no, that just absorbs water. Salivary gland, yes. Stomach, no, that's too acidic, it just produces pepsin. Liver, no. Salivary gland, yes. Pancreas, yes. Small intestine, yes. Let's just look at the last one. Stomach, no. Okay, right. A student investigated, here we go, there's our required practical, the effect of pH, right? So we're looking at pH. So what we're going to have is we're going to have buffers, right? So a buffer solution is kind of like pH 3, 4, 5, 6, 7, 8, and what it does, it stays at the same sort of pH, right? It's kind of like a very consistent pH on the activity of amylase, right? So we've got our spotting tile, right? We need that. There's our spotting tile. Okay. Right, prepare, this is the method used to prepare amylase solution pH 5, right, so we've got pH 5. Mix the amylase solution with starch in a boiling tube. So we've got amylase, right, so amylase goes in, let's start changing the colours a bit, amylase goes in and starch goes in. Okay, remove a drop of the amylase starch mixture every 30 seconds. Right, so what we've got is we've got it on 0, and then we go to 30, and then we're going to go 60, then we're going to go 90, then we're going to go 120, and test for the presence of starch. Presence of starch, we're going to use iodine. Okay, so what it should do, start off with, is at that point there, it should go blue-black. At that one there, it's probably going to go blue-black, and it's only when it doesn't change has the amylase done its job. Record the time when the starch has been digested. Now I think the hardest part about this is that goes blue-black, that kind of goes blue-black, It's deciding when the actual experiment has finished. Record the time. So what we've done is recorded the time when all the starch has been digested, right? I don't know what the time could be. Let's say, I'm guessing 120 seconds. Okay, doesn't really matter. They may well tell us in a minute. Repeat steps 1 to 4. Repeat, right? As soon as it says repeat, I'm then thinking, don't mess in class. Dependent is what I'm measuring during the practical. That's got to be time. In class, independent is what I change between practicals. pH 6, pH 7, pH. So pH. Now, I don't even know the rest of that. Oh, there we go. What was the independent variable in the investigation? Don't mess in class. So what do I change between? pH. God, I've written all that down, and all I had to do was write 2 letters. Describe how the student would know when all the starch has been digested. Right, so what you've got to do there is describe when you know all the starch has been digested. That because you have iodine turns starch, I'm being lazy again, blue, black. And so when all the starch has gone, the iodine stays orange brown, okay, because that's kind of like the colour of what iodine is. Oh my God, look at that graph, a weird looking thing. Okay, so figure 6 shows the student's results, right? Well, okay, let's go with it. pH, time taken to digest all the starch. So time taken to digest. So therefore, if it was quick, then they are good conditions. If it's slow, they are bad conditions. Okay, that sort of makes sense, doesn't it? Right, so let's have a look. So at this point here at the bottom, time taken, right, less than 2 minutes. pH 5, 10 minutes. pH 8, 8 minutes. What was the optimum pH for amylase? Oh, now what I've done is I've kind of messed it up a little bit, haven't I? Because I can't see it. Right, so what you do is you get out your ruler. Right, I'm going to change the colour, I'm going to do it green. You get out your ruler, right, and you start there because that is then obviously the bottom. You get your ruler and you draw a line right straight down. Okay, and you use a ruler, not like I've done because mine's a mess. And then that's 6.5, 6.6, 6.7, 6.8. So the pH is 6.8. You've got to be accurate. If you're not accurate, right, then you're just not going to get a mark for it. Oh, there's another graph. Different. The scientist did a different investigation. Oh, getting crafty. Right. This is the method used to prepare amylase solution at the optimum pH. So that's an optimum pH is pH of 6.8. Mix the amylase solution with starch in a boiling tube. Okay, we did that before. Measure the concentration of sugar every 10 seconds. Right, so 0, 10, 20, 30, 40, 50, all the way up to 120 seconds. Right, however they're measuring the concentration of sugar, I don't know. Right, but there's a graph for it. Time, concentration of sugar in arbitrary units. Very good. Okay, kind of makes sense, doesn't it? So what it does, it starts off at 0, concentration of sugar goes up, 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 and then what it does, it starts to flatten out. How much time did it take for the amylase to digest all of the starch? Right, so what you've got to do then is you've got to look at when it actually got flat. How much time did it take for amylase to digest all of the starch? It will be 82 seconds, okay, because that's then where it actually got to the flat point. Okay, right, now 3.6 is determine the rate of sugar production per minute at 40 seconds. Right, so rate. As soon as you start saying rate, that means it must be something over time, right? Because rate is always per something. All right, so saying there at 40 seconds. So what I'm going to do is I'm just going to put a little purple dot at 40 seconds. So we've got to do the rate there. Now it's saying here also you've got to do the rate per minute, right? So my final answer is going to be arbitrary units per minute. Now what people will tend to do if they're asked a rate question is they'll just go up from 40 and across, and then they'll do the concentration divided by the time. Now that doesn't work, not when you're doing a rate. So what I'm going to do is I'm going to kind of do it per minute already. So if I'm at 40 seconds, I'm then going to kind of do, oh my god, I can't draw a line up here, right? That is 30 seconds to the left of that. And this one, 40, 70, oh my god, right, 40, 10, 20, 30 is 70 to that side of it. Then what we've got to do is I've got to do a tangent. So when I'm then going to do a tangent, I've got to get the tangent so it goes along right through that point there and gets to there. And it's got to kind of be, if I just do it in blue, kind of got to be the same distance away from the line there as it is there. Right? That's kind of the idea of it. Right? That's what my kind of simple little head kind of thinks. Then what I'm going to do is I'm then going to do my triangle like that. And I've then got to work out the rate. So the rate is then going to be the concentration. Now I did this before and that came out as 2.1. Then I'm going to do my time, which is 60. Then to calculate my concentration and my time, what I have to do is I've got to do 2.1 divided by 60, which will then give me my concentration per minute. Now that works out to be 2.1 divided by 60 equals 0.035. Okay, now sometimes the numbers don't work out particularly very well, so because I've done that rate there, then what I've got to do is I'm then going to multiply that 0.035 by 60. That'll then give me my rate per minute, which kind of ironically turns back into 2.1. Okay, question 3.7. Right, so what I've done up here is I've then just put from the mark scheme. So level 1, which means you can get 1 or 2 marks. That's where it is points are identified and stated simple. So it's just simple answers, right? Nothing really kind of constructive or logical. Level 2 is where relevant points are identified and there's a little bit of attempt at logical thinking. It's kind of like your planning really. Level 3 is relevant points, right, and reasons are identified, and there's kind of logic to it. Okay, so if I now look at the question itself, right, I'll start over here. Explain. So what I'm going to be doing there is I'm going to be kind of explaining. So I'm going to do a describe, and then I'm going to do an explain. How the structure of enzymes, that's my first part that I'm going to talk about, is related to the effect of pH on amylase. Okay, so it's on the activity of amylase. So what I'll do is I'll start off with the structure of an enzyme. So an enzyme has a 3D shape. Okay, the whole process, right, that is enzymes that we're talking about, and it is kind of, it follows the lock and key hypothesis. Right, so we all know kind of what that is. Enzymes has an active site that is a specific shape. So what I've done there is I've then talked a little bit about it. Just kind of adding in a little bit more. So starch is the substrate for amylase. Obviously you don't go writing off the side of there. Right, so what I've done then is I've just talked about the structure of an enzyme, okay, and I've done that in then green. Right, so now let's have a look at the effect of pH. Okay, so pH, what you do is you have a graph for pH, so pH 7, and usually what it does, it kind of does that sort of shape there. Well, that might be 3, that might be 11, okay, it's that kind of shape. If you had pepsin, pepsin would be here, because pepsin is then in the stomach, so the pH of 3 or 2 will be its optimum. Right, so pH, so this is then talking about the effect of pH. pH above and below the optimum changes the shape of the active site. So what I'm doing there is I'm looking at the question again. I've done structure of the enzyme. I'm now doing effect of pH and what pH actually does to it. Okay, so then what I've done is I've talked about pH and what it kind of does. The final part of the question is related to the effect of pH on the activity of amylase. So then what happens to the activity of amylase? Now, the substrate can't fit, so therefore amylase cannot work. And that, in an extreme, is called denature. All right, so just recapping it again. The green is I've talked about the structure of the enzyme. In red, I've then talked about the effect of pH. Okay, so what pH actually does. And then the final part of it, I've then done the activity of the amylase. So what I've done is I've kind of addressed every single little part of it. So I've then got the level 3 here because I've done a logical, clear account of what happens. Question number 4. Right, so what we're looking at here now is photosynthesis. Right, as soon as I see photosynthesis, I'm thinking CO2 plus H2O goes to C6H12O6 plus O2. I don't know if they're going to ask that at all. Right, then balance it, just chuck in purple numbers, 6, 6, 6. Right, so there's our photosynthesis equation. Right, let's go back to the question. Photosynthesis is an important chemical reaction in plants. Why is light? Okay, I didn't even put the light on it, did I? So light is there. Right, so light is needed for the transfer of energy. Okay, now it's also because photosynthesis is an endothermic reaction, it needs energy for the actual reaction to happen. Oh, there we go. What is the equation for photosynthesis? So I just then wrote it down, just going through them. Now, because photosynthesis is making glucose, it's definitely not that one. Carbon dioxide, no idea what that is there. 6 CO2 plus 6 H2O goes to glucose, is that one. All right, now if I then look at the last one, I know it's not the last one because a plant doesn't take in oxygen. And also make sure that you read it and you think, tick one box. If you tick 2 boxes, then you're going to get nothing. If you tick no boxes, then that's just silly, because if you have no clue at all, have a guess. 4.3, a student investigated, here we go, practical, the effect of different colours of light on the rate of photosynthesis. Right, so I read that top statement there, and then what I'm doing is I'm thinking bulb, it's supposed to be a bulb, not an eye. Okay, I'll put bulb underneath it so I know what it is. I'm thinking about boiling tube with Elodea in it. I'm thinking about the liquid, haven't drawn that particularly very well at all. Right, so what I've got is I've got the liquid in there. What was it? Sodium, hydrogen, carbonate. Looks like specific heat capacity. Sodium, hydrogen, carbonate. We had a ruler, right? And then what we did is then we had LED light. Okay, let's just think about all the things that we then had. And then what we then is we adjusted the distance between the light and the Elodea. And we did light intensity 1 over distance squared. Okay, so that's just reminding me of it. So the student used pondweed, good. A piece of pondweed was placed in red light, okay, then blue light, then green light. Right, so what I'm doing here is now I'm thinking about my variables. Don't mess in class. So the dependent, what we measure is going to be the bubbles, it's going to be the gas. The independent, what we change between each one, is the colour of the light. Okay, so I'm reading that from the question itself. Each colour of light had the same intensity. Fabulous. We're not going to be doing anything with that.
Describe how the student should make accurate measurements to obtain valid results. So it's accurate means we've got to keep things the same and we've got to make sure we've got numbers and measurements in there. And to obtain valid results mean they are good for the rate of photosynthesis. Okay, so we'll start off. Let's think about the actual method itself. Now, we're not talking about changing the distance, we're talking about changing the light. So let's start off. We've got 50 centimeters. So the light is 50 centimeters from the pondweed. Okay, so what we do now is we're kind of keeping things consistently. In between the bulb and the pondweed, we're going to put a screen to keep temperature the same, right? So what we're doing here is we're looking at valid results. Okay, so there's no other kind of things that are then involved in it. Or use an LED light, where an LED light then doesn't produce the same levels of heat. It produces barely any heat. Make sure that, again, in your methods, what you're doing, you're going through that method that you think of. Equilibrate for 10 minutes. Right, so that means that you have the bulb 50 centimeters away with all the other different things that are kind of going on with the red light, because we've got red light, to make sure that it gets used to it, so the pondweed gets used to it for 10 minutes. We also need to have the same concentration of sodium, you obviously don't do this, sodium hydrogen carbonate in the liquid itself, whether we're doing it in red light, blue light or green light. Then, accurate measurements and what are we going to be obtaining, right? So if we go after accurate, right, bubbles is probably not the best thing to do because bubbles are all over the place. Bubbles are big, small, difficult to count. So the best way of actually collecting our information to get valid results is collect the gas, and for that we could use a gas syringe, which is the best, or just a measuring cylinder. Okay, so just think about what I'm doing here now is I'm describing, right, to get accurate results means I'm going to get a really kind of good number out of the actual results itself, and the fact that are valid, so valid means I'm then keeping out all the control things exactly the same all the time. Time it for 2 minutes. See how much gas, and obviously you doing that, I'm going to be using a stop clock. Then the thing that you've got to just mention all the time is you then record your results. Okay, you record your results onto a table of some sort. Then what you're going to do is you're going to repeat. You repeat using blue and green light, keeping all equipment the same. So you're keeping the LED 50 centimeters away, you're having exactly the same amount of hydrocarbons in there, and what's it called, exactly the same amount of sodium hydrogen carbonate in there, so everything then is kept the same. The only thing you're then changing is the blue and the green light. When you've done that, say it all the time, you've recorded it, then what you've got to do is you've got to do a mean, you've got to do a calculation, you've got to do a graph. Okay, now depending on what the practical is, and probably for the best one for this one, a graph would probably be a bar chart, right? Because a bar chart would then show the difference between the 3 different colours of light. Now obviously my screen there looks like an absolute mess, and what you would have to do is before you actually start writing down what you need to do is think about the order that you're going to do it in. And also don't forget there's extra pages at the end of the exam. Right, so now what we've got is we've got a results graph, okay, a bar chart. So the scientists investigated the effect of different wavelengths, right? Let's just think about wavelengths now, right? So what we've got is that is a long wavelength, that is a shorter wavelength, that is a very short wavelength. So wavelength is from peak to peak, trough to trough, peak to peak, peak to peak. Okay, the wavelength of light determines the colour of the light, right? So Richard of York gave battle in vain, right? So each one of those then has got a different wavelength. Okay. Figure 8 shows the student's results. Figure 9 shows the scientist's results. Right, so we've got another graph that must be coming up next. So figure 8 shows the student's results. So what we've got, so we've got red light, blue light, and green light. So obviously the plant photosynthesis is better in the blue light. Okay, so photosynthesis is better in the blue light there. Green light, it barely does anything at all, right, which is kind of weird. So if you think about that, there's your leaf, right, typical leaf, right, obviously. Okay, my artwork is as usual spectacular. If I've got red light shining on a green leaf, right, what happens is the red light is absorbed. If I have blue light shining onto a green leaf, the blue light is absorbed. If I have green light, oh, why am I doing a G, right, green light on it, the green light is reflected, that's why we see it as green. So therefore, that kind of explains a little bit about why green has very little rate of photosynthesis, because the actual green light is getting reflected off the leaf. Don't know if that's relevant or not, that's kind of just what I'm thinking when I see this question. Oh, that's a wiggly one. Okay, so this is then the scientist's results. So rather than us having a bar chart, right, that's got just kind of individual things that are then going on it, right, where it's just either this or this, this now looks like the wavelength where there's much more variety in it, right? So what they've done is they've then used 400 nanometers, 410, 20, 30, 40, up to 500, up to 700. So they split it up a lot more than us just saying red, green, blue. Okay, so why are the results for the two investigations presented differently? Okay, so the colour of light is discrete. Okay, which means there's lots of wavelengths. Okay, or you could be writing down wavelength is continuous. Right, so what I've then done is I've then said that what the investigators have done is they've done it as like a continuous thing. But if you notice in there, why are the results of the two investigations presented differently? I've only then said that the scientist results are discrete, or they've done it discrete, where the wavelength is continuous, where the student has picked only one wavelength per colour. So what they have done is they've just got a bulb, where the scientist, he's then got much more accurate equipment, so what he can do is they can present it differently because he's got a full range of the wavelengths. Suggest a range in wavelength of green light. Okay, now I should have the graph on there really, right, but what the graph looked like, it looked like that, going along, and it kind of at the top there, and it went down the bottom here, did a bit of a wiggle around, and it went up there like that. Because this is rate, I can then see that here and here, there and there, where it's dead low, is the wavelength for green light. So I'm then going to put in there 475 to 650. Now there is kind of a little bit of a bump in there, I think, somewhere. So what you could have done is you could have done there to there, or there to there as well. Okay, question number 5. This question is about tumours. Okay, describe the similarities and differences between benign tumours and malignant tumours. All right, the question there tells you exactly what you've got to do. All right, describe means you're going to be saying facts, and you're going to do similarities, differences between benign and malignant, So in a way, the similarities, you're going to be talking about both of them. And the differences, then you're going to say benign does this, malignant does this. So what you're doing is you kind of set it all out for you, as long as you know what a benign and a malignant tumour are. So what I'm going to do here is I'm going to write down the similarities are, I'm even just going to bullet point it, are they both have a change in DNA. They both have uncontrolled growth. They both create a lump, and they both have abnormal cells. Okay, so that is then where I've said the similarities are, or similarities between benign and malignant tumours. They both have a change in DNA, they both have uncontrolled growth, they both create a lump, and they both have abnormal cells. Okay, then we can do the differences are. Now this is then when you've got to be a little bit more careful, right? Because what you've got to do is you've got to say a benign tumour is slower. They don't invade local cells. They can't spread. And they don't have secondary tumours. Okay, now remember this is a describe question. So what you're doing is you're describing the difference between them. A malignant, God I can't write today, a malignant tumour, do the same sort of things that was obviously before, is faster, don't know why it's faster to grow. It does invade the local area, because what it does, it kind of produces like little roots that sort of go into the local cells. It can spread, God, and it does produce secondary tumours. Okay, but the key to this then is looking at the actual question itself. Describe means I'm telling facts. Similarities, I've done that in purple at the top. The differences, I've then done benign tumours do this, malignant tumours do this. So I've used the question to write out my answer. Well, there's a graph. Right, figure 10 shows data about skin cancer in males and females for different age groups in the UK. It shows the number of new cases of skin cancer in 1 year, so number of new cases, and the number of people with skin cancer per 100,000 population in 1 year. Right, so it's a double-sided graph. So new cases is the bar chart, and per 100,000 is then the line graph. Okay, and what you're going to be looking for on this is you're going to be looking for the trends. I don't know how they're going to ask it, right? It looks quite interesting there that it kind of crosses over. You can see that that dotted line there is steeper than that one. You can see that at this point here, 65 to 69 for, who is it? Male cases is the highest, and you can see that male cases are higher right the way up there, and you can see that female cases are higher when they're at the bottom. Now, there's loads and loads of bits of information that you can pick out on that, and now it's going to be what they're actually going to ask you about it. There are no new cases of skin cancer diagnosed in people younger than 15. Okay, no, none at all. No skin cancer cases for people younger than 15. Right, so think about that logically. Right, if you think about it logically, what you can have is people under the age of 15 have little exposure to UV light or ionizing radiation. Okay, even though you lot have, right, up to the age of 15, right, it also hasn't had time for mutations to happen. Okay, so this question is it's kind of like the 2-mark question. It's all about the fact that you haven't had that exposure to UV ionizing radiation and even things like other carcinogens, like smoking. Okay, so even though carcinogen smoking isn't a big one for skin cancer, it kind of shows you that you haven't had the exposure to these things. And it also takes time for the cancers to actually develop and mutate. Now, 5.3, give 2 conclusions about the number of new cases of skin cancer. Number of new cases of skin cancer, and this is then our bar charts. All right, now there's absolutely tons of things that you can put on there. Number 1 that I could then write is new cases, so up to, let's have a look at females, up to the age of up to 59, women have more new cases. Okay. Another one that I could maybe have is more males than females have new cases over 60. Okay, so that's kind of what I talked about before, is that's then when you've got your grey bar, or whatever colour it is bigger for males, and below is then bigger for females. There is then the fact that most are diagnosed, or most people, or most numbers of people are diagnosed between 65 and 69. Okay, so most people are then diagnosed, so this one here, that's where the most is. And then with males, the most are diagnosed 65 to 69, but then it drops off. So what you could do is, there's absolutely tons of different things that you can actually pick up on that graph. 5.4, the data for the number of people with skin cancer is given per 100,000 population. Suggest why the data is not given as the total number of people. Right? So, I mean, if you think there's 50 people, right, living in a country, and you say that there are 4 people with skin cancer, and then you've got a country that's got 100 million, and you say there are 200,000, or however many there is, it just doesn't, it's not comparable. So what you have to be able to do, you have to be able to account for different size groups, so you can compare. So whenever they do it out of like a population, like 100,000 population or whatever it is, it's the whole point is so that you can compare. It's almost like a bit like taking a mean. It's taking a mean number per 100,000, so it is a comparison. Right, now the graph came up again. Okay, so this time the graph is then coming up, right, which then means there's going to be another kind of analysis. Now we've already talked about the number of new cases, so I suspect now it could be this. I might be wrong, I might not be. Describe two trends shown in figure 10. Use only the data for the number of people with skin cancer per 100,000. So we're only using that data there. Right, so what we're now doing is we're looking at the trends, so number of people, so we're looking at the lines. Okay, there's a pattern. Number of cases, increases with age. It does. Low here, high here. Okay, that's a general pattern. Females, I'm just going to write up the bottom here, increase with, God, I can't write, with a steady rate. That's right from the start. So if you look at the female line, right, the kind of the darker line, it's just like a consistent line, right, that just keeps on going, just goes at kind of standard rate. Males, rate increases round about here, age 55 to 59. That's like another pattern. Age 55, male plus female are the same. That's that point there. Okay, there's quite a lot of different things that you can actually look at there, right? You can also talk about the difference in the end, how there's a big discrepancy between the distances, where at the lower part, the discrepancy isn't a huge amount. You could also talk about the fact that females, up until the age of 55, have got more per 100,000. The estimated population of males aged 80 to 84 years was 694,000. Calculate the number of males aged 80 to 84 with skin cancer in that year. Use figure 10. Right, so for this one, what you need to do then is you've got to calculate the number of males aged 80 to 84 with skin cancer in that year. 80 to 84, I'm going to go up and I'm going to go to here and I'm going to read across, and I can see that that is 116. So 116 people per 100,000 have skin cancer. Right, so I'm going to do my sum, 116 times by 694,000, divide it by 100,000 to kind of spread it out a little bit, and my answer will then be 805.04. Fabulous. Okay, so I've got my answer there, 805.04. Spectacular. Right, there's all the marks. Let's just keep reading. Give your answer to 3 significant figures. So what I've then done is I've just thrown a mark away, because obviously that isn't 3 significant figures. So to 3 significant figures is 805. Question number 6. This question is then about the heart. Okay, so immediately what I'm doing is I'm then thinking structure of the heart. Okay, I'm thinking about the route red blood cells take around. I'm not going to draw a heart, right? What I could do, I could draw my square that I always draw. Lungs there, I could put body there. So what happens is it starts in the, go on, which way round is it? That's my left, so that is right ventricle. It goes out to the lungs, it then goes back to the left atria. It then goes to the left ventricle, then it goes round to here to the body, and then back round again to the right atria. This, because it's to do with the lungs, is the pulmonary artery, because AOA goes from the lungs there. That is the pulmonary vein, because it's to do with the lungs. It comes out of the left ventricle, so that's the aorta, to the body, and then it goes back via the vena cava. And then you've got valves here, so I'll do that in a different colour, valves here, and you can't even see that, valves here, and you've got valves here. Okay, well, that's kind of the main structure of the heart. Why is the heart described as an organ? Okay, completely irrelevant to what I've done then. So let me think about that path. We've got cell, we've got tissue, we've got organ system, and then we've got organism. Right, so that's kind of our cell structure, isn't it? The organisation of something. Right, so why is heart described as an organ? It's because the heart is a group of tissues with specific roles. Okay, because an organ is made up of tissues. Right, there we go, structure of the heart. Okay, there's our heart diagram. All right, so let me just think about it then. I'll think about my square again I've got over there. So that one then will be, it's not the left, so it's the right atrium. Right, then that there, no, what am I talking about? Right ventricle, right atrium. The vessel that goes into the right atrium is the vena cava. That there is then the left ventricle. That there is the left atria. The one that comes into that is the pulmonary vein. We've got the left ventricle, so pumped out of the left ventricle is the aorta, and then going into the right atria is the vena cava. Right, so I remember my order. Now let's see what the question says. Valves in the heart keep blood flowing through the heart in one direction. Okay, it'd be a bit stupid if it then do all sorts of directions. Figure 11 shows the heart with one of the valves labelled. There it is, so it's between the left ventricle, the biggest part of the vent, and the aorta. That sounds like a problem. Explain the effects on a person if the valve labelled figure 11 developed a leak. Right, explain. So firstly what I'm going to do is I'm going to describe it. So blood would go back into the left ventricle. Now we're not talking about all of it, we're just talking about small bits. Right, so that's kind of how I described. Then we've got to do is we've got to think about at least 3 or 4 different reasons why that's then going to cause a problem. Because if you've then got, what colour shall I do it in blue? If you've then got blood going back into here, that then means that there's less blood going down the aorta, right? And what's the point in that? So therefore, there will be less oxygenated blood in the body because there's less oxygenated blood. So therefore, there will be less aerobic respiration. That would then mean that the person is not getting enough oxygen, so therefore out of breath. They'll have less energy because they can't get enough oxygen. Lactic acid will build up and they'll probably be very tired. So just think about it. If you're not getting enough oxygen around the body because it's not getting out of the heart and it's not going out the aorta, describe it first and then explain is then what the consequences are then going to be of it, right? So then it's talking about there's less oxygenated blood, which then means that this is then going to happen. 6.3. Okay, so what we've got here is faulty heart valves can be replaced using biological, living, or a mechanical heart. The faulty valve is replaced during an operation. It sounds like a big thing, right? You've got to go into the heart and replace the valve. Biological valves, let's do this in green, are from animals or humans. Allow blood to flow through them normally, so really what you're doing is you're replacing what was already there, but they can wear out and stiffen over time, so might need to be replaced. Okay, that's a bit of a pain. A mechanical valve are made from synthetic materials, may cause blood clots on the surface of the valve, that doesn't sound good, require anti-clotting drugs, so that sounds like something that you're going to have to take for your whole life, oh yeah, and can last a very long time in ideal conditions. Okay, so that's a good thing, isn't it? A young woman enjoys extreme sports and would like to start a family. Okay, the woman needs a heart valve replacing. Describe the advantages and disadvantages for this young woman of having a biological heart instead of a mechanical. Advantages, disadvantages, we can forget all about really the mechanical valves because we're not really going to be talking about, because we've got to think about the advantages and disadvantages of a biological heart. Okay, so we've actually got most of the information in the question itself. So an advantage is it allows blood to flow normally. Another advantage is, because it says on this one here, that requires clotting drugs, there is a reduced chance of clotting. Pregnancy, there'll be a reduced chance of bleeding in pregnancy. So what I'm doing is here I'm using the question. Don't need anti-clotting drugs, because what it does, it says in this bit here, that requires anti-clotting bloods, but up there it doesn't. Because there's less clots, then I'll go back to red, less risk to fetus. Oh, I can't spell. To fetus, so when the baby's growing. Disadvantages, let's do those in purple. It could be rejected. Okay, so if they're made from animals or human donors, right, there is a chance that they're going to be rejected. So therefore, might have to take immunosuppressants. You've also got the fact that might have to be replaced. And also, it might need, an animal might be killed to replace it. And then from a human, it's obviously going to be somebody that's already passed away, right, who's then donated their things. Now, with this question, it's 4 marks, right? So it's basically you've got 2 advantages, 2 disadvantages. But what you need to do is you need to look through the information. And all the answers that you ever need for this, right, are in the question itself. Allows blood to flow through normally, right? So the advantage is that the blood flows through normally, where with a mechanical valve, it doesn't flow through normally. They could wear out and stiffen, which means they're going to be replaced, right? So that is obviously a disadvantage. Look at the information that they're talking about and just analyse it. Now, that is then the paper 1 higher GCSE for biology. And what you need to do is you need to be able to just look at a question and kind of have a train of thought as you're actually going through it. And if you can do that, then the questions are then going to kind of flow, right? Graphs, as you can see from this paper, graphs are absolutely vital